All right, now we're going to get to do something fun and a little bit more practical. We're going to talk about blood typing and why it's so important to match blood, especially when people are in need of blood. To do that, though, we got to have a couple definitions. I'm going to want you to be able to define what an antigen is versus an antibody and the process of agglutination. We're going to look at these two systems, the ABO system and the RH system. And then we're gonna wrap it up by talking about the RH system, specifically in a disease called hemolytic disease of the newborn, and talk about how it's caused and how you can prevent it. All right, so first of all, antigens are, first definition, where this word comes from. Really, if you take anti and gens, gens means to generate things, and anti in this case is not used for the meaning not, but we're really talking about antibodies. So antigens are things that generate the production of antibodies, and antibodies are what your immune system is producing that recognizes foreign invaders like bacteria and viruses and goes after and attacks them specifically. In this case, it's going to be different though, because your red blood cells actually have antigens on it, and they're not foreign, but your body's going to make antibodies to determine blood cells that aren't yours. So antibodies are secreted by lymphocytes, and you learned about lymphocytes being a type of a leukocyte, type of white blood cell. And these are the things that are going to be responding to all those foreign invaders. They look like a little Y. We're going to talk about them more specifically with the immune system lectures. But for your purposes for today, just know that these antibodies that are produced by the lymphocytes are going to go after and attack things that look foreign to them. So let's start with this ABO blood typing system. We name the type of blood, either A blood or B blood or AB blood or O blood, and it's named for the antigen that's on there. So all these red blood cells have antigens, which are little proteins that are sticking up out of the red blood cell plasma membrane there. And depending on what you get from mom or dad, you're going to get one genotype from mom and one genotype from dad, and they're going to come together. So you could either get an A from mom and an A from dad, and you would have A antigens. Or if you got a B from mom and an A from dad, you'd have A and B antigens. So you can get any combination of them. They can also be shooting blanks, meaning they may not give you any antigens. So there's 4 basic combinations that we're going to look at here. First one is if you get antigen A. Well, antigen A in this case looks like a little lollipop. So it doesn't necessarily look like that, but we're just going to call it that for this demonstration period. So if that blood cell is lined with antigen A's, here's what the really fascinating part is. Your body is going to make antibodies for the type of antigens that you don't have in this system. So if you have type A antigens, then your body is going to make type B antibodies that are going to attack type B antigens. So we call these anti-B antibodies because these antibodies are going to go attack type B antigens because you don't have them, and those would be foreign to your body. So you produce antibodies for the antibodies that you don't have. Let that sink in for a second, and we'll look at just the opposite. If you have type B blood, then you're making these type B antigens. So if you make type B antigens, it probably makes sense that you would make what type of antibodies? You'd make anti-A antibodies, and they would go attack any cells that look foreign to them. If type A blood got mixed in with your blood, those antibodies would go attack them. Well, you could have another combination. You could get A from mom and B from dad, and you'd have type AB blood, which means you've got both of the antigens on your red blood cell. If you had both the antigens on your blood cell, then your body's not going to want to attack itself. So your body is not going to make any anti-B or anti-A antibodies, because if it did, you would be attacking your own red blood cells. And then you've got just the opposite extreme. You might not get antigens from either your mom or your dad, in which case you would just have a plain old red blood cell. That's me. I've got no antigens on my red blood cells that are in this A or B class. So I'm called type O. Let's put this in here. Type O. And it's not that you have O antigens on that red blood cell. It just means that you have nothing on there as far as A's or B's go. So in that case, what do you think is going to happen? Your body's going to recognize that, oh, if A or B come in here, they're going to be foreign. So I'm going to make both anti-A and anti-B antibodies. That's me. So let's kind of look at how this thing is classified. There's a little chart in your book that kind of puts all of these together. I just want to explain it to you because it's a little confusing if you happen to go look at it. So the genotype is talking about what you get from mom and dad. So they give one example of type A blood, and here's another example of type A blood. And what they're saying is this. You can get type A, you can get type B, or they're giving this I here a letter meaning that you got nothing. Zero, nada, no antigens. So they're giving you example. Your genotype, if you had mom and dad on here, you could get A from mom and A from dad, and that would give you your phenotype. Phenotype is just what is expressed. This would be your genotype here. These are your actual genes that you have. Phenotype means that you're going to have type A blood. In which case, you already know that your antibodies, you're going to have anti-B antibodies. So type B blood, you could get a B from your mom, a B from your dad, and you would have phenotype B, and you'd be making anti-A antibodies. Now, what they are showing you on here is something that's a little bit different. I didn't say it with this first one. They have this AI. All that that's saying is that you can actually have A from one parent and a blank from another parent, and you're still going to end up with type A. With type B, you could get a blank from one parent, so we'll make that an I, and a B from another parent, and you're still going to get type B. So that's what these guys are referring to, just the little blanks. If you had an I from mom or a blank from mom and a blank from dad, then you would get type O. If you got an A from one and a B from another, that would give you type AB blood. That's all that this graph is showing you, and showing you the resulting antibodies. So here you'd have 0 antibodies, and here you'd have anti-A and B antibodies. Okay, so let's look at this. Well, what's the big deal about having antibodies that are going to be attacking the opposite cells? So here you've got antibodies that are attaching to corresponding antigens. So in this case, you've got a cell that's type A, and if this type A blood is given to somebody who has type B blood, we know that those type B blood people are going to be making anti-A antibodies. So I'll call this anti-A antibodies. So if this type B person is already making anti-A antibodies and we give them type A blood, then these antibodies are going to hook on to the antigens of type A blood. And this is where it gets nasty. because those antibodies can clump on to different red blood cells. So now it's going to trap these two red blood cells together, and when it does that, we call that agglutination. And it looks like, well, it's not that big of a deal, but if you think about it, you're going to have a bunch of red blood cells all around here. And if you have all of these antibodies hooking on to the red blood cells, what that's going to do is that's going to make a big clump of red blood cells. And when those red blood cells clump up like this is what we're calling agglutination. Those red blood cells can rupture more easily because they can't get through the capillaries. And so they're going to get squeezed to death and they're going to lyse. And when they lyse, they're going to take those 250 million hemoglobin molecules, and that's going to get spilled out into the bloodstream. Well, if they spill out into the bloodstream, all this hemoglobin and the heme groups in it, that's going to cause kidney failure. And even these red blood cells clumping up aren't going to allow the kidneys to filter the blood. And so that's going to be really problematic. And with kidney failure, you can die pretty quickly. So this is a result of agglutination. We can type somebody's blood by purposefully putting anti-A or anti-B antibodies into a blood sample and see if it clumps. So in this case, we've got type A blood here. And what we've done is we've added some anti-B antibodies to that blood. And anti-B antibodies aren't going to do anything to type A blood. So it's going to look kind of like this. But if we happen to add type A antibodies to the type A blood, then it's going to clump up. And this is what it looks like when it clumps up. You see all this clumping of red blood cells there. Similarly, if we took type B blood, we took two different stains of it, and we added type anti-B antibodies to the type B blood, we're going to see all of this clumping going around here. This clumping is what's going to cause those red blood cells to lyse, as opposed to if we just added type anti-A antibodies to the type B blood, nothing is really going to happen. There's no agglutination going on in here. That's looking just fine. If you have type AB cells, and if you add anti-B antibodies to it, of course they're going to clump. And the same thing with anti-A antibodies, they're going to clump. Remember, this is adding antibodies to test it. This isn't adding someone's blood to somebody else's blood. So you got to kind of separate that in your mind. That's the only way it gets confusing. This is just, think of it in a lab. You're trying to identify someone's blood. Maybe you're a crime scene detective and you're trying to identify someone's blood type. So let's look at this next blood typing system. This is called the Rh blood typing system, and it's based on an antigen we call the D antigen, or the Rh antigen. And we call it the Rh antigen because we first found it in rhesus monkeys, and rhesus starts with a Rh. So we call it the Rh antigen or the D antigen. Either one works fine. What's interesting about this antigen is that you don't make antibodies against it unless you've been exposed to the antigen. So people that are Rh negative that don't have this antigen, they'll cruise along just fine and never knowing that they had it. Only if their blood mixes with somebody else's blood who's positive, then they'll start to make the antibodies against it. If you're Rh positive, clearly that your body won't be making Rh antibodies because you won't want to be attacking your own blood. Now, what's kind of interesting is that most people are Rh positive. If we look on this little chart here, it kind of breaks it down. The phenotype means what's actually expressed by different ethnicities. And you can see that for the most part, most people have an 80% chance or more of being Rh positive. In fact, Caucasians have the least amount, and that's about, 82%, 83% of white non-Hispanic folks are Rh positive. When you get into the Asian and African countries, 90% or more of people are Rh positive. So only a few really are Rh negative. And why is that so bad? Well, there's a condition called hemolytic disease of the newborn. And this occurs when mom is negative and dad is positive. And it has to be this specific combination of mom being positive and dad being, mom being negative and dad being positive. Well, guess what? I'm positive. So I, Professor G, is O positive. And my wife just happens to be negative. I think she's AB negative, something like that. So here's what can happen. You have a baby that's growing inside the mama's uterus there. That baby can be Rh negative or Rh positive. Obviously, mom can't contribute the negative or the positive gene to it, but I could, and I either got the positive from my dad or from my mom or from both of them. We don't know. All I know is that I have the gene. So my alleles, meaning the different types of genes that I could give, would either be positive or negative, potentially. So if that baby happens to get my positive genotype, then this baby in here is going to be positive. Now, the good news is that the red blood cells don't cross the placenta. So that baby's going to grow in here just fine, and the mom's never going to know that the baby has these positive D antigens, and she's not going to go trying to attack it because she's never been exposed to D antigens before, and she still isn't exposed. So the trouble comes during labor. And during labor, you have a lot of tissues getting torn, and there's bleeding, and you could have some of this cord blood, or blood from the baby here, get exposed into the mom's system. And at that point, the mom's going to say, oh, uh-oh, foreign invaders, got to start making a bunch of antibodies against these D antigens. Well, the baby's going to get delivered, and the baby, this happens during labor, so the baby's already on its way out. It's probably separated from the placenta a little bit, and the baby's going to be born just fine. No worries about it. But the mom now has antibodies in her blood. So the second baby that's born, if that second baby is Rh positive, so I'm going to write down here, if second baby is Rh positive, and it's only if that baby's positive. If the next baby's negative, any baby negative after that, no problems. Mom's not going to attack that baby. But a future baby, if that baby is Rh positive, then the mom's antibodies can actually cross that placenta because they're small enough that they can go across that placenta. And those antibodies can attack the red blood cells and cause them to agglutinate. And that usually doesn't happen until closer till, I think, in like the third trimester, usually, or the baby could actually be born, but it'll have those mom's antibodies in it, and at that point, the baby may only survive a couple of weeks or so. And this is a very, very sad condition. Let me just show you another slide here that kind of shows you the same thing. You've got positive dad mixing with a negative mom, and in that mom, you're now going to have a positive baby. So mom doesn't make any antibodies until that baby is born, and the mixing of all that blood gives the mom all these antibodies. So the next time that there's a positive baby, those antibodies are going to go attack that little baby. Fortunately, there is a medication that we can give, and that medication is called RhoGAM, and the mom can take it. I believe she can take it right before she gives delivery and right after she gives delivery, and that prevents the mom from making any antibodies against that D antigen, that Rh antigen. And so if the mom never makes any antibodies, everything will be good moving forward, and all of her future babies will be okay. And that was the case with me and my wife. She got the RhoGAM shots, everything was fine. But I'll tell you, it just because we know how this works now doesn't mean that people make mistakes or are immune from it. I had a lady in my last class was telling me that she just found out that I think it was her cousin or a friend of hers possibly, um, didn't get the RhoGAM shot and the doctor overlooked it. She was Rh negative. I don't know if they knew what the father's blood type was, but in any case, if you're Rh negative, it's probably a good idea to at least ask the doctor about getting the shot because it's the simplest thing to prevent loss of life. And if you learn one thing out of this class, to save a life the easiest way, make sure you or if a friend of yours or a family member is pregnant and they have Rh negative blood, make sure that they're following up with that RhoGAM shot. And that ends this part of the lecture.